Welcome to Discover with Dr. Dan, the Proactive Health Podcast. Today we are talking about the science behind antibodies and their usefulness, not only related to COVID-19, but also in other medicinal applications. With us on the show today is Professor Shawn Owen, an antibody expert from the University of Utah. Welcome to the show, Shawn.
Thanks for having me, Dan. It's great to be here.
Yeah, pleasure talking with you. By way of introduction, Shawn Owen is an assistant professor in pharmaceutics and pharmaceutical chemistry and adjunct professor in the departments of biomedical engineering, of medicinal chemistry, and of internal medicine at the University of Utah. He is also a member of the Huntsman Cancer Institute Experimental Therapeutics Program and the Nano Institute of Utah. Dr. Owen's research focuses on antibody-based therapeutics and diagnostics. He is an expert on the construction and analysis of antibody-drug conjugates and is the co-inventor of a patented antibody split enzyme system for diagnostic applications. He's published over 30 research papers on drug delivery and biotechnology, and his work has produced 4 additional patent applications. Dr. Owen is recognized with several prominent University of Utah teaching awards and is sought for outreach workshops by both academic and industry sponsors. Again, listeners, you can see Professor Owen, very well versed in antibodies. And again, Shawn, you're a friend. We've known each other for over 20 years, and it's great to have you.
Thank you. Thanks for that introduction. I will say, just for the sake of truth, the way to win teaching awards is to tell a lot of jokes and give out a lot of A's.
There we go. I like it. So, Shawn, let's dive right in. When we think about antibodies today, especially in today's climate, people think about COVID vaccines. So tell us a little bit about the different COVID vaccines that are on the market today and how they work.
Yeah, sure. I'll go into a little bit of detail with each one of those and how they relate to antibodies. So the antibodies that are most relevant is the antibodies that your body is producing. So what you want from a vaccine is to train your immune system to be able to fight an infection. Mm-hmm. And so in the case of COVID-19, you want to train your body to be able to block that infection when it eventually, hopefully, doesn't come. But when it does come, it'll be able to block that. And so the different vaccines that are out there, the ones that you've probably heard the most about are Pfizer and Moderna. Mm-hmm. And those are the 2 that have been approved in the United States for use, and those are the 2 that are being administered here in Utah and across the nation. Both of these are actually a brand new technology. These are the 2. This type of a vaccine has never been broadly used before. Okay. And the way these work is a little bit different than a classic vaccine. So just for context, a classic vaccine, the way it works is you take a piece of the virus that you're looking at to block, and you usually will attach that to something else to help stimulate the immune system. And then you'll give that as an injection to a person. And so when the body sees that, they process that fragment of the vaccine and then present it to the body and trains the body so the next time it sees a fragment like that, it will remove it. It'll actually build antibodies against it so the immune system will recognize it the next time it comes around. Mm-hmm. What's different with these new type of vaccines, these mRNA vaccines, is instead of putting a piece of the vaccine, you actually put a piece of the vaccine's code, the mRNA, into the body. And what happens is this actually gets incorporated into our cells, and a piece of the vaccine is actually produced on the surface of our cells using that mRNA as a code. And when that happens, after that, it's the same kind of process. The cells, your immune cells are always kind of surveying the landscape, and they recognize that this new piece on this cell is not normal. It's not part of me. Uh-huh. And then it goes through that same process and develops an antibody against it. So the next time it sees that protein again, it will recognize and fight against it, and it'll launch antibodies against that invasive species.
Wow, that's really interesting. It's cool that a lot of people think vaccines are all the same, and it's interesting to hear what you're saying, that they're different. There's different technology that we can use in order to get the same type of response, get to the end game.
Yeah, I think these ones are really exciting. The technology that has enabled this has only been around for probably 10 or 15 years. Prior to that, we weren't able to use an mRNA to encode. It wasn't stable. The formulations wouldn't work. So it's really exciting that we're able to process this so quickly, and we're getting such great responses so far.
Yeah. So Going along with that, responses, how successful do scientists envision these vaccines to be based on the initial research that's coming back from clinical trials?
Yeah, that's a really interesting question. And it's one that's hard to digest short-term versus long-term. When you'll hear different numbers that are reported in the news. So Moderna or the Pfizer vaccines are 95% effective. And what that means is what they did in the clinical trials, they gave it to a whole bunch of people. And then some people got a placebo, so just a blank version of the vaccine. And some people got the actual vaccine. And then they sent those people back out into the real world. And then they waited for some people to get infected.
Okay.
And then they counted and they said, of the people that were infected, how many of them had the vaccine? How many of them had the placebo? And it turns out that 95% of the people that were infected had the placebo. And so when they say it's 95% protective, that's what it means. Okay. So that there's a lot of caveats there. Does that mean some of the group were more careful than the others or not? It's hard to say what that means. But the way I would really think about it is, let's compare it to things that we already know about and how effective they are. So the flu vaccine that we get, most of us get yearly, that is reported to be about 65% effective. Using the exact same criteria. So that means that it's not, in a side-by-side comparison, the preliminary data that we have, so this first initial result that we have, it's 95% compared to 65%. So that's really, really exciting. I don't know if those numbers are going to hold. We have to test that in a much larger population. But as we start to see those numbers come back, even if we lose, 10% and are down to 85% efficacy, that's still way better than the average flu vaccine. So there's a lot of hope that these things can kind of continue the way they've started.
Wow. And do you think that higher success rate is due to the new technology that they're using here?
I don't know. I would like to say yes. I think as scientists, what we do is quickly form an opinion, and then we test it, and then we poke at it and see if it holds up to it or not. So if you ever ask if a scientist is opinionated, absolutely, we're probably the most opinionated people, but we're also kind of forgiving that we can be convinced a different direction. Right now, the other vaccines that are starting to be out there, like Johnson & Johnson's vaccine, is showing much more normal. It's based on the old technology, and we see about 65% protection, which is close to the flu vaccine, which is on that same technology. So first initial rounds, mRNA is looking really good. And if this is true, it could make a major difference in other types of vaccines that we couldn't do before.
Wow. Very cool. So let's now talk about safety. There's been a lot of talk going around in a public domain, infertility and these sorts of things with these vaccines. What's the real science behind that in regarding to safety and some of the things that are going around?
Yeah, I think that probably we need to break this down into two questions. Is the new technology safe, or are vaccines safe in general? I think that a lot of people have heard the idea of the connection of a vaccine and autism. And unfortunately, that goes back, and it can all be traced back to one single researcher and presenting two different papers. And if you look at the science that went behind that, it was really flawed. And what I like to look at with science is, okay, like I said, I form an opinion quickly, but then I keep poking at it. And with the vaccines, the initial reports that came out that vaccines cause autism, there looked like some correlation, and we can accept that. But then as it's been tested over and over, was it a specific ingredient in the vaccines that was problematic? Well, that's been completely removed from vaccines. So that can't be the issue. And more importantly, I think that we've demonstrated so many times that just because one person thought they saw a correlation one time, of the other 100,000 people that don't see a correlation, that really says to me that vaccines are one of the safest kind of therapeutics that we have. Now, one caveat there, as a pharmaceutical scientist, we always like to talk about toxicity. And it turns out one of the things we repeat quite often is the idea that toxicity is in the dose. So it's how much is given. So really, everything is toxic. If you think about it, water is toxic. And I'm not talking about drowning. I'm talking about if you drink too much water, you can die. So it's really how much are you taking? How is it administered in the right way?
Mm-hmm.
Now, all these things. I mean, vitamins could be toxic if they're administered in the wrong way.
Mm-hmm.
So we want to make sure that we're getting them the right way, right formulations, and really controlling those things. So now with this new technology, so I would say that I've really put that to bed. Standard vaccines, they're safe,
Mm-hmm.
and especially considering the dose that we're getting, they're amazingly safe.
Mm-hmm.
There are some allergic reactions. Almost all of those are because of the extra ingredients that are in those vaccine
Mm-hmm.
formulations. For current vaccines, for the mRNA, I was one that when we first heard about this, I really wanted to dig deep and figure out, are these things safe?
Mm-hmm.
Because it's a new technology. I told you how it expresses on a cell surface. This is completely unique.
Right.
Some of the best studies were actually done about 10 or 15 years ago in dogs. And they administered the same kind of a vaccine to produce a fluorescent molecule.
Mm-hmm.
So they could really easy track this throughout the body. And it turns out that if I was going to be worried about a reaction, it'd either be instantaneous. So as soon as you inject it, your body rejects it and you have some kind of shock.
Right.
That doesn't happen. Or for a long-term reaction, if this protein stays around a really, really long time, then it can start to develop other kind of responses. That didn't happen with the animal experiments 15 years ago. It hasn't happened with the recent experiments that have happened that they've been tracing in humans. So I don't think that any kind of safety questions are a problem. Now, the last thing I'll say, Dan, is there are some people that are having an immediate reaction, and that looks like it's probably from one of the ingredients that's in there. So if you are a person that reacts to vaccines, be cautious with that.
Well, that's interesting. And I think a lot of people don't know that in a vaccine, you have a lot of different carriers and adjuvants and other sorts of things that help in the delivery form. So wonderful. So let's now talk about antibodies. We've been talking a little bit about antibodies in relation to these vaccines. You're an expert in the field of antibodies. How did you get into this field?
Yeah, it's one of those kind of serendipitous possibilities that every once in a while happens in your life. I was working at the University of Toronto, and my job was, one of the projects that I was assigned to work on was to connect something like a vaccine to an antibody. And so my job was to stick these two parts together. And that's really what I'm good at is linking two molecules together. And at that time I started working with a professor that helped with some of the newest types of antibodies that are out there. I got really fascinated with these molecules because at that time I only thought of them as an extra molecule that I could use. They could let my vaccine or my molecule go to a specific place.
Right.
But it turns out they can do a lot more than that. And they really have a lot of complex structure. And so just really became fascinated with them and decided when I started my own lab that part of that lab would be dedicated to understanding antibodies, new versions of antibodies that are coming out, and how we can really harness that power, that natural power.
Wow. Yeah, in the literature I've read, it seems like we're just scratching the surface on the applications of antibodies and how they can be useful, not only in medicine, but overall health applications.
Yeah, I agree.
Nice. So when people think of antibodies, at least in today's climate, they also think of COVID-19 tests, right? You have the traditional test, which most people don't know what it's called, but it's an RT-PCR test. And then you have the rapid antibody tests. So tell us about these tests, how they work, if they're effective, those sorts of things, that there's a lot of confusion. And then I know that you and your group are doing some interesting things when it comes to antibody technology in relation to COVID-19 testing and, diagnostics in general.
Yeah, thanks. So there's three types of tests for COVID out there. So the first is what you just mentioned, this RT-PCR. And what this is it's looking, remember I talked about at the very beginning, this idea of the RNA, that's the code of the virus. So that's what that test is looking for. It's taking a little bit of a blood sample or a spit sample and testing to see if they can find the code of the virus in there. The second type of test is called an antigen test. And so an antigen is what your body is looking for. So that might be a fragment of the virus itself. And so this kind of there's a lot of rapid tests looking at looking for the antigen. Mm-hmm. So some of the ones that are used by the NBA when they were all stuck down in Disneyland, Disney World, that was an antigen test looking to see if they could find fragments of the antibody. And a lot of those are kind of like a pregnancy test where you have that strip that'll give you a kind of a plus or minus, but usually doesn't tell you how much is there. The last kind is the antibody test. Now, this is an interesting test because it takes time for your body to build up antibodies against a virus. So if I'm infected right now and I just got infected, I probably wouldn't have antibodies against the virus. Mm-hmm. So testing early on, there only would be one kind of antibody, but over time, as your body really builds up that immune response, it shifts to a different kind of antibody called an IgG. Mm-hmm. And so this antibody is the one we really, really want. And so each of these have different values. You can see that maybe how long it's been since a person was infected, how much virus is there. All of these tests can be useful at different times. And the antibody test will really be useful, especially if somebody's been given a vaccine, 15 months later, can we check their blood and see if they still have those antibodies around? Are they still protected against the virus?
So it seems like these tests that are out there, they have their time and their place and their application depending on what you're tracking and what you're trying to see and the time frame.
Yes, exactly. And also kind of the viral load, like how much infection is there. Sometimes an antigen that's looking for a piece of that vaccine, there might just not be enough, a piece of that virus, that it might not be enough there to really tip the scale to say it's positive. Okay. But a PCR might be more sensitive and be able to pick that up. Okay.
So we have these tests that are out there, and I know you and your research group and other groups around the world are, developing antibody tests. If we already have tests out there, why do we need other ones, and why is new technology needed?
I think this is a great question, and I think part of it is market driven. I think part of it is access. A lot of these rapid tests require kind of an instrument that was developed by a company. And so do you have access to that instrument or not? Is there an instrument at every clinician's office or not? And also processing those samples. So is it something that a nurse could do, or do you need a specialized individual to process those samples? I think there is a real saturation, and it's become kind of the wild, wild west in diagnostics right now. Everybody has a COVID assay. Mm-hmm. Some of them work really well. Some of them don't. I think the reason we need new technology is especially because we need to focus in on some of these really minute questions like level of the virus, how much is there, or what my lab is working on is really focusing on those antibodies that are protective. Okay. Sometimes these are called neutralizing antibodies. So my test that we're developing, it will not tell you probably in the initial stages if you're infected. Okay. But after you've been infected, maybe after you've recovered, do you have antibodies that will protect you from future infection? Or if you've been given the vaccine, have you then developed these antibodies? And that's really what we're really interested in is can we develop a test that's rapid that also will tell you the levels of antibodies that you have. Okay. So you can say with confidence, yep, okay, the vaccine worked and I'm protected.
Okay, interesting. So again, these tests have applications, especially down the road, right? If you've been infected and then you want to track efficacy and infectivity, I guess, for lack of a better word, down the road.
Exactly.
Okay, so we've talked about antibodies and antibody tests related to COVID. Let's talk about antibodies and other applications, whether it be diagnostics or medicinal applications. What does the future look like there?
I think that antibodies probably hold as much promise as for our generation as things like blood thinners and cholesterol inhibitors did for our parents. Like you mentioned earlier, we're really just scraping the tip of what these things can do and how useful they are. And a big reason that they're becoming so much, we're being able to access them more, is how antibodies are generated. So it used to be if we wanted to get an antibody, we had to do it the natural way, which means we would take an animal, we would inject them with some antigen, and we'd wait for them to develop an antibody, and then we would try and match that. But now we can do this all chemically. So we can do this essentially just in a dish. We can generate our own antibodies. The other big difference is understanding that we actually don't need the full antibody for some of the things that we're trying to do. We can actually use small parts of it. And this became out because we recognized that other animals, other mammals, and even sharks have different types of antibodies that are still effective. And now we've been able to kind of manipulate these so that we can even use them in humans for therapeutics and for diagnostics. I think that the real power comes in we're able to do some things that we can't do with small molecules that we can with antibodies. We can block some proteins. We can, a really good example that I always bring up with people is most people have probably seen a commercial for Humira, which is a treatment for plaque psoriasis. And that kind of treatment, there are some small molecules that work, but what happens with plaque psoriasis is there's a signal from your body that is overstimulating your immune system. And it's a large protein called TNF that's moving through your body, and trying to get a small molecule to block that is problematic. But using an antibody, a large molecule to block to a large antibody, it can be very, very specific and only block that TNF. So the specificity is better, and also, the magnitude can be quite a bit better. I thought it would be interesting to tell your listeners a little trick that they can use in their and impress their friends. So there's a nomenclature, there's a naming system for antibodies. And if you watch, the next time you watch a commercial, they'll always say the trade name and they'll say the generic name of these medicines. And what you're looking for is MAB, M-A-B at the end of the name. So Humira is adalimumab, M-A-B, monoclonal antibody. And so the next time you hear this MAB at the end of an announcement, you can just turn to the people in the room and say, that is a monoclonal antibody. And just impress everybody with your vast therapeutic knowledge.
All right. It's good to know. So we've talked a little bit about antibodies, how they work, application in COVID, COVID diagnostics, vaccines, those sorts of things. So tell us, where do you think the future of antibodies is going, specifically related to me and the listeners, right? Why should we care? And how can this research that you and others are doing benefit my life?
I think that this is a great question, and it's one that I actually reflect on a lot because of how much we care about antibodies. One of the real powers of antibodies is that they are very, very specific. So a lot of times when you take a cancer drug, we look at all these horrible side effects that come from cancer drugs. And the problem is because those drugs go everywhere in the body and they interact with a lot of other parts of the body that we don't want them to. They might affect the liver. They might affect, cells in your scalp that are causing hair growth. But with an antibody, because of how they're made, they only will bind to a really specific target. So even though they might go somewhere else in your body, they won't interact with that. So there's some really powerful antibodies that are now being used as therapeutics for cancer. So Herceptin is one that a lot of people know. This is used for breast cancer, and it binds really specifically only to the tumor cell. And maybe if there's that same antigen in other places, it might bind those, but the vast majority is only binding to that tumor cell. And so another thing that my lab's really interested in is, so that antibody binds really specifically, but we really want to amplify the effects that are happening as a therapeutic. We really want to kill that tumor. So there's a whole new generation of therapeutics called antibody drug conjugates. And what these are antibodies that have been fused to a small molecule, a really potent molecule. Now, normally, if you just injected that potent molecule, the same problems that you normally see with chemotherapeutics, it'd be toxic. But if you put it on the antibody, you got to stop and think, well, where's it going to go now? Well, it's only going to go, it piggybacks off that antibody. So now you get the bonus of the targeting of the antibody and that Trojan horse, that amplification of the drug. So, I mean, heaven forbid that any of us ever get cancer, but the hope that in the future we now have better therapeutics that not only are going to be more efficient at killing the cancer, but have less side effects than some of the traditional therapeutics. And I think this is untapped, and we're starting to see that same kind of approach being moved into infectious diseases. Things that are really, really bad, like MRSA and other, other problematic infectious diseases.
So what you're saying then, in that example, using antibodies and drugs, you're able to get better specificity of hitting the target. And when you get better specificity, does that mean in general the side effects can be less?
Exactly. So a lot of toxic effects they're called off-target toxicity. And so if these drugs they get to another place, or they interact with another molecule or another protein in your body, they can cause that toxicity that we don't want. And so we really want to be able to kill, but only kill what we want to kill. But I think that is kind of limiting our thoughts. I think that we could go beyond that. Okay, we can kill cancer. Maybe we can kill an infectious disease. But what other things could we do? What if we could turn things on? What if somebody, instead of turning things off, instead of killing things, what if somebody is not producing some kind of an enzyme or some kind of a protein that they really need in their body? Well, what if we flip that around and now we deliver a therapeutic that turns on the protein? Now we don't want that turned on everywhere throughout the body, but we only need it in the pancreas, or we only need it in the liver. And maybe we can use antibodies to really, amplify the effect or turn on an effect that we really want.
Wow. How cool. It's interesting to see all the different applications, right? Again, when people think antibodies in this day and age, they think of, COVID testing, but it's amazing to see the frontier and the wide open applications here.
Yeah, I think one of the other thing that's really exciting is how we can create antibodies now. It used to be that if we wanted antibodies, we had to actually harvest them from an animal.
Right.
Or even now we hear a lot about plasma that's being donated by people, right? And that's still a great way to get things when we don't have a synthetic antibody. But now almost all the antibodies, once we know the sequence, we can create them and we can produce them just in cells in an incubator. So we can avoid having to use animals. We can avoid having to, have donors to get these things eventually. I will say, though, we have a major limitation with antibodies right now, that if somebody out there has a good idea, I'd love to hear it. Antibodies are large proteins, and your body is designed to digest proteins. So there is no pill formulation of an antibody. You can't take these by mouth. They're all going to be an injection. And that would be an immense hurdle. If we could figure out a way to make the convenience of an oral that then you could take and have the power of an antibody, that would solve some major problems.
Wow. Very cool. All right. I'll let you know if our listeners give any comments of how that can be done. So that's interesting. So there are some hurdles to overcome. There's a lot of research that needs to be done, which is a good thing if you're in the field that you're in, right? A lot of good opportunities there.
Yes.
Awesome. So Shawn, this has been great. Sure appreciate the great insights that you've given our listeners. My mind has been blown as we've talked. It's been amazing. What final thoughts would you give our listeners? We've talked about a lot of things. Is there take-home messages or final thoughts you'd like to give?
Yeah, I think that I like to come back to that idea that as scientists, we tend to form our opinions really quickly. But one thing that we remain healthy skeptics, even on our own opinions. And I think people need to remember that, scientists don't know it all. We come up with an idea and then it changes over time. I can't think of a single idea that I've worked on in my lab that at the beginning was what it was at the end. It always changes and morphs. I think that, remember that is a process. The science is a process. And so when we look at things like COVID and we'll say, it used to be the policy we don't wear masks and now we do. Why is there such flip-flopping? Well, our understanding changes. What we know changes. And so we need to be receptive to that. And we need to continue to grow as we get more information, and be willing to change as we get that more information, or you're going to be stuck in a really bad place.
Wow. I love that. As a scientist myself, Yeah, I love that, science is a journey, right? A lot of people think that science is set in stone, right? That it's, the same today and yesterday and forever, whatever, but it's great to hear you say that, science is a changing discipline, and it's all about, seeking the truth, but it's a progression.
Yeah, it's a process. It's iterative. Sometimes it's painful, but I think that eventually we learn more and more, and it's exciting to see the discoveries that we make so quickly.
Awesome. Well, thank you again, Shawn. We know you're busy running your research group and doing a lot of things. Grateful that you take time out of your day to talk with us.
Hey, thanks a lot for putting something like this together, Dan. I really appreciate it. Hopefully It was useful.
It totally was. Thanks to our listeners. I hope you found today's information fascinating like I have. And again, I hope you found some actionable information that you can do and some knowledge regarding vaccines and new applications to help us be healthier. So thanks for listening. This is Dr. Dan signing off.